Okay, you guys, we're going to go ahead and move on in the 5.2 notes. In this video, we're going to look at topic 2, which covers something called sex-linked genes. And so in these notes, we're looking at lots of different ways that genetics can sometimes be a little bit more advanced than the simple model that Mendel came up with. This will be another example of something that's of non-Mendelian inheritance, something that Mendel didn't quite figure out that occurs sometimes for certain traits in organisms. And so I'll kind of explain what this is, but I want to start by talking about this guy. His name is Thomas Hunt Morgan, famous biologist who studied the inheritance of traits in fruit flies, which are called Drosophila. This word right here, Drosophila, are these fruit flies that you see right here at the top. And so he did a lot of different crosses involving these fruit flies and tracked lots of different traits in these fruit flies, a lot like what Mendel did with his pea plants, but now we're using these flies instead. And he actually made some very important discoveries. One of the biggest discoveries that he made, and this was in the early 1900s, very early on in the 1900s, so about like 50 years after Mendel figured out a bunch of his stuff with his pea plants. One of the biggest things that he figured out is that genes are located on chromosomes. He was actually the first person to figure that out, that there's these chromosomes inside your guys' cells, and the information for all of your traits are actually located in those chromosomes. Like that's where all of your genes are and your information is for all of your traits and characteristics and phenotypes. It's all there. And so these alleles that Mendel came up with actually exist as pieces of information on these chromosomes. He's the one that made that connection, which meshed really well with what Mendel came up with, the fact that you have 2 alleles for each gene, and when you produce offspring, you're only going to pass on one of those alleles, and there's this law of independent assortment where whatever allele you give for a certain gene has no effect on what allele you give for another gene. It matched really well with a lot of the principles that Mendel had already previously discovered. Now knowing, once this guy finds out that, oh my gosh, these alleles and traits and this information is all located on your chromosomes, which, like I said, makes a lot of sense with stuff that Mendel figured out. So for example, you have 2 sets of chromosomes, a set of chromosomes from your mom and your dad, and they're each carrying a bunch of genes on them with instructions for certain traits. And so for example, like you could have a chromosome from your mom that has the instructions to have round seeds, and on another chromosome that you got from your dad, there could be the instructions to have wrinkled seeds. Like you're going to have 2 alleles because you have 2 sets of chromosomes, one from your mom and your dad, which that would give you a heterozygous genotype. You'd be big R, little r. And then for seed color, right, you could have the instructions on a chromosome that your mom gave you has the instructions to have yellow seeds, but then on another chromosome that your dad gave you, a homologous chromosome, it has the instruction to have green seeds, right? Those would be homologous chromosomes, each carrying different versions of that gene, in this case yellow and green, which would make you heterozygous for that. So in this case, like this is a cell that would be a dihybrid, right? Like they're heterozygous for 2 different traits, big Y, big R, little r. And that genotype and the 2 alleles that they have for each of those genes is directly connected to their chromosomes. Like it's their chromosomes that are carrying this information. And then during meiosis, those chromosomes will duplicate and make copies of themselves and then line up in the middle and be pulled apart and ultimately packaged after 2 rounds of division into these daughter cells that each have 1 copy of each of those homologous chromosomes, each carrying a single allele for each gene. And so you can see that there's different combinations that are possible of how these chromosomes that carry the Y gene are packaged with these chromosomes that carry the R gene. And you could end up with these combinations out here, which matches what Mendel was saying about the law of independent assortment. Like whatever R you pass on has no effect on what Y allele you pass on, and that all combinations should be equally possible. With the way that meiosis works, that's exactly what's going to play out. We talked about this a lot in class already. Anyway, he figures that out, which is huge in the history of biology. But then he also does a lot of other things. And so I want to talk about some of his basic experiments that he does with these flies and the crosses that he does. And so like I said, he's going to ultimately figure out that these genes, the instructions you have for certain traits, are located at specific locations on these chromosomes, which we'll talk more about soon. But anyway, I want to talk about the basics of his experiments, kind of like what I did with Mendel. to explain how he discovered something called sex-linked genes, which is a little interesting. So in his experiments, Thomas Hunt Morgan, he worked with these fruit flies, and he specifically focused on what's called wild type phenotypes and mutant phenotypes. Wild type phenotypes are like the normal phenotype that's found in the majority of organisms, individuals, and mutant phenotypes are these certain phenotypes or certain versions of traits that you find every now and then in certain random individuals. And so for fruit flies, most of them, the wild type version is red. Red is very common. Most of the flies have red. This is the normal version. So wild type means normal. Think of like that's the normal phenotype for fruit flies. But every now and then you come across a fruit fly that has this mutant version of eye color where it's white. And so for a lot of the traits that he was looking at a wild type normal version versus a mutant type version. And what he did when he did these crosses is he figured out that sometimes you end up with ratios in the offspring that don't match what Mendel would have said. And so I'll kind of show you. So here's a good example of like the gist of some of his experiments. He would start with, and you're going to see a lot of similarities between this and Mendel. He's going to start with true breeding flies. So these are flies that have a certain trait where basically true breeding just means that they're homozygous for that trait. They're always going to pass on a certain allele for that trait. In this case, this is a true breeding red eye fly. And so it's always going to pass on a red eye allele because it's homozygous for red eyes. And this is a true breeding white eye fly. And that's because it's homozygous for the white eye allele. Anyway, he's going to start with those guys and he's going to cross them. That would be the P generation. He's going to produce the F1 generation. The F1 generation, you have the hybrids, which come from these two opposing true breeding flies. And those hybrids all had red eyes. Now, so far, this is perfectly consistent with what Mendel would have said. So here it looks like we have a case of dominance, where red is dominant to white. Like this fly is obviously going to end up with a red allele from this true breeding parent and a white allele from this true breeding parent. This kid's going to be heterozygous, but have red eyes because red apparently is dominant. So far, that's all working out the way Mendel would have thought it worked out. But then what Morgan did is exactly what Mendel did. Morgan's going to take these hybrid flies in the F1 generation and he's going to cross them with each other. So he's going to cross these heterozygous flies with other heterozygous flies to produce the F2 generation. Now, according to Mendel, this is what you would expect to happen. If you have a heterozygous fly that has a red allele and a white allele, and you cross it with another heterozygous fly that has a red allele and a white allele, you should get this outcome in the offspring, where 75%, a majority of them have red eyes, and about 25% of them have white eyes, if everything works the way it's supposed to. If you have these two alleles, you can only pass on one of them, and all these combinations would work out. But what happened is that is not the outcome he got necessarily. So what he actually found is that overall, he did get a 3-to-1 ratio of flies having red eyes and white eyes. But what he noticed when he carefully observed these offspring is that the phenotypic ratio was different for the male flies versus the female flies. So what he noticed is that 75% of the overall flies had red eyes, but every female always had red eyes. Like 100% of the females always had red eyes. And it wasn't until you looked at the male flies where some of them now had white eyes. And so when you looked at the male flies, there was a 50-50 split. Half of them had red eyes, but the other half had white eyes. So overall, it was a 3-to-1 ratio. But among the females, it was actually just 100% red. And among the male flies, it was 50-50. It was a 1-to-1 ratio, which that is very odd because according to Mendel, that shouldn't happen. Like these phenotypes should have nothing to do with your sex. If you're born male or a male or female fly, like this should have nothing to do with that. And so, what Morgan concluded from this is that this gene for eye color must be located on a chromosome that has to do with the sex of these flies. Apparently, there's something going on. There's some connection between the chromosome that controls the sex of these flies and this gene for eye color. That's the only explanation you could think of for why you would get a different outcome among the male and females. And that's exactly what was confirmed, is that this gene for eye color is located on a sex chromosome. You guys have 23, for humans, we have 23 pairs of chromosomes, but one of those pairs is a pair of sex chromosomes, which are X and or Y chromosomes. And so in humans, there's two types of sex chromosomes. There's X chromosomes and there's Y chromosomes. Females are born with two X chromosomes. They have an X chromosome and X chromosome for mom and dad. Males are born with an X chromosome from one parent and a Y chromosome from the other parent. And they get the X one from their mom and the Y one from their dad. And that's what makes them male. And so that's the way these sex chromosomes work out. And so what he figured out is that the X chromosome, that this gene for eye color must be on the X chromosome. And when you're looking at the females, it's going to play out differently than how it plays out in the males, because the females are always going to be born with two X chromosomes, which means they're going to get two sets of instructions, two sets of alleles for eye color, which is actually normal, right? For every gene, you're supposed to have two chromosomes that carry that gene, a homologous pair, one from your mom, one from your dad. You're supposed to have two alleles for all of your genes. Well, for females, if it's a gene on the X chromosome, that's normal. Like, you do have two X chromosomes and you do have two alleles for that gene, for eye color in this case. But if you're male, something different is going to happen. If you're male, you actually only have one X chromosome, and then you have this other Y chromosome. And the Y chromosome doesn't have the same genes as the X chromosome. The X chromosome has hundreds of genes on it for humans, but the Y chromosome is like this really tiny chromosome. It only has a handful of genes on it that relate to male development. And so they're not a true homologous pair. So whatever gene is on the X chromosome, you don't have a second copy of it on the Y chromosome. They're actually different chromosomes. They don't have the same set of genes on it. So for guys, if you're born with an X and a Y chromosome, you actually only have one allele for every gene on the X chromosome, because you don't have a second X chromosome. And so that's going to be kind of definitely way different than what Mendel said, because he was saying you have two alleles for every gene. But now we're going to have a situation where sometimes for some genes, if you're male, you only have one allele because you only have one X chromosome. And so I'll show you guys some examples and how this plays out in terms of crosses that we might do and Punnett squares that we might have to set up. But that is kind of what this picture is showing you here with these flies. And so here's an example of a sex-linked gene involved in a cross. And so when we do a cross involving a sex-linked gene, we have to keep track of the sex of the individual, if they're male or female, because we need to know if they have two X's or if they have an X and a Y. And so what we do is we write the X and the Y if they're male, and we write the two X's if they're female to represent the two X chromosomes that the female has and the X and the Y chromosome that the male has. And then for the allele that we're tracking that's located on the X chromosome, we write it as a superscript. And so let's say we have a red eye male. Well, a male has an X and a Y chromosome, right? And on the X chromosome, let's say this gene is sex-linked. Sex-linked means it's on the X chromosome, this gene. And so the eye color gene's on the X chromosome. The allele he has on this X chromosome would have to be for red eyes, which they're representing as a capital W, because that's the normal version. In this picture, I guess I don't like that they chose W, but they chose W. And so a big W represents red eyes. And then they have a Y chromosome, but on that Y chromosome, there's no allele for eye color. The gene for eye color isn't on the Y chromosome. So they only have one allele, and it's on this X chromosome. In this case, it's for red eyes because they have red eyes. Now, the female, she has two X chromosomes, which means she's going to have two alleles. On this X chromosome that she has, there's an allele for eye color. And on the other X chromosome she has an allele for eye color. One came from her mom, one came from her dad. And since she has white eyes and white is recessive, she must have two white alleles, two recessive alleles. Now, when you cross them, the rule plays out the same way, where you can only pass on one of your sex chromosomes to your offspring. So if you're a female, you have two X chromosomes. Well, when you produce eggs through meiosis, those are going to separate, and you're only going to pass on one of those X chromosomes or the other X chromosome. Like when you make eggs in your egg cells, there's only going to be one of those X chromosomes. You only pass on one. Same thing with males. You have an X and a Y chromosome. Well, when the male makes sperm cells, the sperm cells will either contain the X chromosome or the Y chromosome, but not both. You only get to pass on one of them when you make sperm cells. So because of that, we can set up a Punnett square to show, okay, what are different ways that mom could pass on one of her X's or the other X's, and what are different ways that dad could pass on his X versus his Y's, and how could this be inherited in the offspring if they have to inherit a sex chromosome from mom and a sex chromosome from dad. And so you set up a Punnett square, a 2x2 one, and you can put one of the parents, let's say dad, on this side here. So dad has an X chromosome and a Y chromosome. He can pass on one or the other. If he passes on the X chromosome, that's going to carry the allele for eye color. And then mom has two X's. She can pass on one or the other. In this case, both of them have a white allele. And so here, this shows you the different ways those can be combined in the offspring. So like this offspring right here could get an egg cell that has this X chromosome and a sperm cell that has that X chromosome. So now this fly would be heterozygous. It would have two X chromosomes. One of them has the red allele and one of them has the white allele. And this would be a female fly because it has two X chromosomes. Or you could end up with this fly, same thing with this fly over here. It could end up with this type of X chromosome from dad and this X chromosome from mom, and it would end up being born female and be heterozygous. It would have a red eye allele and a white eye allele. And because red is dominant, it's going to have red eyes, just like this fly has red eyes. But then you can have a situation like this where this kid gets an X chromosome from this mom's egg, but then a Y chromosome from dad. So now this kid is only going to get one X chromosome that came from mom, and the Y chromosome has to come from dad. And so now this kid will be born male, and it's going to have a white allele, and that's it. It only has one allele, and that's on that X chromosome. So he has white eyes. Whatever allele you have on that X chromosome, well, that's going to be your phenotype because you only have one allele. Same thing with this kid. It's going to get that X chromosome from mom with the white allele on it and the Y chromosome from dad. So this kid will be born male and have white eyes. Okay, so that's how you do these Punnett squares. We'll do a couple examples together in a moment. But basically, you got to keep track of the X and Y chromosomes. So now you're going to write an X and a Y to represent those two different chromosomes. And on the X chromosome, you're going to write as a superscript what allele is on that X chromosome. And for females, you're going to have two alleles because you're going to have two X chromosomes. And for males, you're going to only have one. And then when you pass on your chromosomes to your offspring, you can only pass on one of your sex chromosomes. Just like any homologous pair, you can only pass on one or the other, carrying one of these alleles or this other allele. So anyway, now so you can see here in this outcome, you have a 1-to-1 ratio of flies with red eyes to white eyes. But if you look carefully at the different sexes here, if you look at just the female flies, 100% of them have red eyes. All female flies always have red eyes. And if you look at the male flies, 100% of them have white eyes. And so you get a different outcome between the males and the females. Anytime you have a cross and the phenotypic ratio in the males doesn't match the phenotypic ratio in the females, that is usually explained by a sex-linked gene. So for some reason, the sex you were born is influencing how this gene was inherited, which can almost always be explained by the fact that that gene must be a special gene that's located on a sex chromosome, usually the X chromosome, because the X chromosome is a big chromosome with lots of genes on it. And so for the huge majority of questions we do, it's usually always the X chromosome that has the gene. But that would explain why it doesn't necessarily get inherited the same way or the same outcome amongst the male and the female offspring. And so that's what all of this Morgan figured out from his experiments and his crosses that he was doing, that some genes are located on sex chromosomes, or in this case, the X chromosome, which we call that a sex-linked gene. And so let's do some examples, actually. I'll pull up my my camera with some of these questions here, and I'll show you how this plays out. It's actually not tricky. It's pretty simple and straightforward what you got to do, as long as you're keeping track of everything correctly. Moment to plug this in. Like for this first question, it says colorblindness is a sex-linked gene. Cross a woman who is a carrier for colorblindness, which is recessive to normal vision, and a man who has normal vision. What is the probability of having a girl that's colorblind? What's the probability of having a boy who's colorblind? So there's a gene that controls whether or not you can see certain colors correctly. And there's different severities of this gene, but for this specific type of colorblindness, it's red-green colorblindness, and has to do with how the individual how their eyes perceive red and green wavelengths of light, and sometimes it's they don't see it the same way as someone who normally would see it. And that has to do with a gene that's located on the X chromosome. And so color blindness, though, is recessive. So if you have the normal allele, having normal color vision is dominant. And so this says that there's a woman who's a carrier for color blindness. Carrier means that this woman is heterozygous. It means that she doesn't have color blindness, but she's carrying the recessive colorblind allele. But because it's recessive, she's not colorblind, but she's a carrier. So it basically means she's heterozygous. So for the woman, she would have 2 X chromosomes. And on each of those X chromosomes, she would have an allele for color blindness. And so you would have to figure out what 2 alleles those are. And like I said, it's going to be heterozygous because she's a carrier. So she has a dominant allele, which is normal vision, and a recessive allele for color blindness. We'll use the letter N just because normal is dominant and it's easy to keep track if it's uppercase or lowercase. So she has a dominant allele on one of her X chromosomes and a recessive allele on another X chromosome. And we're going to cross this woman with a man who has normal vision. Now, the man will have an X and a Y chromosome, and on that X chromosome, there's going to be an allele for colorblindness. Well, it says he has normal vision, so he's going to have the dominant allele. And then on the Y chromosome, there is no letter. He doesn't have an allele for colorblindness on the Y chromosome. The Y chromosome doesn't have this gene. So that's the genotypes you're starting with. This mom, she's heterozygous, 2 X chromosomes. Dad, only 1 X chromosome, and he has the normal vision allele on it. Okay, now when you do the cross, you're going to set up a simple Punnett square, just 2 by 2. And so mom, she has these 2 X chromosomes, but she can only pass on 1. When she produces egg cells through meiosis, only 1 of these X chromosomes will be in each egg. Some of them will have this X chromosome that's carrying the allele for normal vision, and some of them will have this X chromosome that has the recessive allele for colorblindness. And then same thing for Dad, he can either pass on in his sperm the X chromosome that he has or the Y chromosome. So some of his sperm, half of his sperm should have this X chromosome, the other half should have the Y chromosome. And so here are the different ways that egg and sperm can meet. You could have this egg and this sperm joined together to make an offspring, which would produce this outcome. A kid getting 2 X chromosomes, 1 from mom, 1 from dad, in the egg and the sperm, each carrying these 2 alleles. Okay, or you could get this kid here who gets 2 X chromosomes, 1 of them carrying the dominant allele, 1 of them carrying the recessive allele. Or you could have this kid here who gets an X chromosome from mom's egg, but a Y chromosome from dad's sperm. Or you could have this kid who gets an X chromosome from mom's eggs and a Y chromosome from dad's sperm. And so here are your different possible outcomes here for this cross between this woman and this man. And so if you look here at these kids, this would be someone who's born female, and they are homozygous dominant. They have 2 normal vision alleles, so they have normal vision. And this is a female that's born. And then this would be another person who's born female, and they're heterozygous, a carrier like their mom was, but they have normal vision because normal is dominant. This would be someone who's born male, and they have normal vision because that's the allele they have. They only have 1. And this would also be someone who's born male, but this person would be born with color blindness because they have the recessive allele. And so the question is, what is the probability of having a girl that's colorblind? Well, if you look at the possible daughters that are born, no one born female is going to have color blindness. So we can say 0% chance that you're going to have a colorblind female born from this cross. And then it says, what's the probability of having a boy who's colorblind? Well, if you look at the individuals born male, they, some of them are going to be colorblind. There is an outcome that leads to a colorblind male. And so if you figure out the probability, this is where I get slightly different answers. Sometimes some students say 25%, some students say 50%. And this is when you have to read the question very carefully. So this question is saying, what is the probability of having a boy who is colorblind? So because the question is being asked in a way that's asking for the probability of having a boy who's colorblind, the answer would be 25%. So we would say a 25% chance that they're going to have a colorblind male. And that's, now if the question said, like, let's say this couple, they were pregnant, having a kid, and they found out it was going to be a boy. Like, the moment you know it's going to be a boy, then it's no longer a 25% chance that they're going to be colorblind. Once you know it's you're going to have a boy, then these outcomes are no longer possible. Like, so that kid who's going to be born male either it's going to be one of these kids. Now it's a 50-50 chance. But the way this question's written, it's written as if we don't know yet, because they want to know what's the probability of having a boy who is colorblind. Well, then out of all possible outcomes leading to boys or girls, colorblind or not, there's a 25% chance to be colorblind. So that's how you guys do these type of crosses. There's a couple other questions you guys can try out. We'll do lots more practice in class, but hopefully that mostly makes sense so far. Anyway, that's it for topic 2. Thank you guys.